Good morning, everyone. It's a pleasure to be opening this year's OTS meeting, which I'm sure is set to be like another outstanding success for the history of the society. Welcome to the educational session. This year we've had the chance of hosting a slightly different session, where instead of diving deep into a specific scientific topic, we're going to be learning about how scientific discoveries can be translated into commercial products. Both Alan, the session's co-chair, and myself are scientists turned entrepreneurs. And the theme of our session today, which is innovating and commercializing within the next generation of oligonucleotide therapeutics, essentially crystallizes the mission that we're on, our collective mission, which I'm sure it's something that it's shared with many people within this audience today. The oligo field has had innovation at its core, and we know that this has been both underpinned by academic and industrial advances. And I think as it matures, it's more clear that it's not enough to merely make transformational, groundbreaking technologies and therapeutics. We need to be able to translate those into patients, into things that really matter. So to discuss these and other topics, we've lined up a panel of remarkable individuals, all of whom have a common thread, which is that they are key founders and stakeholders within the field of RNA therapeutics. But what really sets them apart is the diverse backgrounds that they've had and the unique paths that they've taken to be part of the RNA therapeutics field. And we have really very much intentionally curated a panel to showcase the richness, the diversity of experiences, backgrounds, and journeys that can converge into an entrepreneurial journey. So as they share their stories, experiences, and insights, I hope you get inspired by this diversity and that it encourages you to embrace your own path within the RNA oligonucleotide therapeutics field. So just a bit of admin on how the session is going to be run. We won't be taking questions after each of the presentations, and instead what we've allowed is plenty of time at the end of the session to have a panel Q&A discussion. So please do keep your questions towards the end. Let us know what are the topics that you would want to dive deeper into. And we'll be taking both questions from the live audience and the online one as well. So we're really looking forward to have very constructive and enlightening conversations today. I think they will provide some tangible advice to those of you who are looking to take the route of entrepreneurship, but also be very informative and helpful for everyone in the audience today. So with this, I'd like to introduce our first speaker, who is Marcin Kozelewski. He's a professor at City of Hope and within the immuno-oncology department. And he's also a founder of Duet Therapeutics. So
Good morning, everyone. It's a pleasure to be here in Barcelona for the first time, and thank you for the introduction, Anna. And let me give you a warm-up presentation, which is dealing with the very early stages of deciding about startup formation. I'm still continuing to be an academic scientist, so these are my disclosures. And very briefly about academic lab at City of Hope, which is dealing primarily with cancer immunotherapy, but we do employ oligonucleotide therapeutics, which turned out to be excellent for purpose of targeting immune network at the cellular as well as molecular level within the tumor microenvironment. And we focus primarily on targeting antigen-presenting cells, which are at the beginning of this cycle of immune response stimulation that have been characterized and shown in this famous scheme from Chen and Mellman. But we focus on the stage which requires maturation, full activation of these antigen-presenting cells, which are really information processing hub in the immune system. And that can be more effectively done with targeting intracellular transcriptional regulators in these cells rather than by targeting multiple surface molecules or single immunostimulatory pathways within these cells. For this talk, we primarily focus on the STAT3, where we develop technology that have been later licensed, and STAT3 is one of these molecules, a master regulator of immune suppression within the tumor microenvironment that really is a nodal point of immune suppressive network responding to multiple stimuli from the microenvironment, translating into multiple effector molecules that can drive immune suppression further. So this role of STAT3 is basically allowing it to inhibit progression of immune system development, immune response development at multiple levels, from antigen presentation to recruitment of T cells and their activation. So these studies started with findings that came across during my postdoctoral work, and then we followed up when starting my own lab with additional findings that basically taught us that just blocking STAT3 by itself is not enough, like immune stimulation using this immunostimulatory CpG DNA is also not enough for long-term anti-tumor immune responses, and you do need a combination of these two effects to make this effect long-term and persistent and engage T cells. And then we followed up with basically a strategy that gave us a proof of concept for using oligonucleotides in a cell-selective manner for blocking STAT3. So there was a first oligo which employed STAT3 siRNA and targeted it into antigen-presenting cells using a single piece of DNA, targeting it to TLR9 positive cells, which turned out to work really well in vivo, getting to the right type of cells, macrophages and DCs in mice, and then leading to anti-tumor effects in multiple tumor models. So we spent some time trying to understand processing of these molecules intracellularly, finding out, and I think that was the most important part of this, of these studies, that the fact of immune activation allows us also to get out of the endosomes quickly, so within a couple of hours after stimulation in TLR9 expressing cells, we could really get out the siRNA into the cytosol and make it work. And then we spent a couple of next years optimizing this strategy for targeting human cells, and then eventually made a switch and decided to use antisense, a molecule specifically for targeting solid tumor microenvironment. Turned out that this composition of a single-stranded DNA with a single-strand antisense molecule had a better penetration into solid tumors. Nonetheless, the mode of action of these molecules remained the same. They escape from early endosomes through TLR9 activation, allowing the STAT3 knockdown, and that unlocks the potential of immune activation and antigen presentation by these cells, allowing us to jumpstart immune responses. So over this time, there have been a couple of inflection points, like that we can call, when we started thinking about building up the startup, and the first one was around 2009, with coming with a first publication on a proof of concept for using the CpG STAT3 siRNA oligo, and that turned out to be really bad timing because of the economic slowdown in US post 2008 crash. So we have went back to the lab and basically entered this valley of death, which is also for academic researcher valley of impact factor, because it's very difficult to generate during this time high impact publication that would still give you enough fuel to fund your research using federal resources like R01 grants or DoD grants. So at this point, we have fortunately acquired private funding, also funding from foundations like Prostate Cancer Foundation, as well as intramural funding from City of Hope, to be able to move on with preclinical studies that involve basically manufacturing at larger scale, toxicology and safety studies that are IND enabling and allow you to move forward. On one hand, also to attract startup and to think about startup formation and attracting investors. So within about 9 or 10 years, we have moved from basic research with these resources towards scaling up of the synthesis of the first 2 molecules of CpG STAT3 inhibitor family. We have completed IND-enabling toxicology studies in 2 species for 2 of those molecules, filed IND and opened a clinical trial for CpG STAT3 siRNA in B-cell lymphoma. So part of these studies gave us enough fuel to seriously revisit the idea of startup formation. I formed together with John Rossi at City of Hope as a co-founder a startup called IDSTAT, which eventually evolved to become Duet Biotherapeutics, which is now focusing on moving these molecules towards the clinical application. So going through the decision-making process, I think it's important to look at some pros and cons at each stage. Academic life and academic career has this huge advantage of giving you freedom and independence in pursuing different research goals, jumping from one target to another. This is, of course, only as possible as much as the funding levels allow, and in context of cancer research, the funding from NCI, for example, is very limited, and the 8 or 10 percentile of grants can be actually funded. So there's some limitations here. Mentoring can be very refreshing and a great experience at this level, but given the funding level, it's very difficult to retain the best trainees and offer them long-term positions, especially faculty or tenure positions. Overall, the tenure track in the academia, which in the past was more stable, it now at least offers some predictability, which can be a benefit as well. But it comes, of course, with additional cost of multiple distractions, administrative tasks, committees and reviews to do. And finally, we have in academia, you have access to fantastic resources for doing in-depth research, which might not be possible with early stage startup, but still, given the focus on basic research, the clinical translation of your findings is relatively slow. So that is in contrast to the startups which have this singular focus on well-defined goals, which allows them to move really fast. It does require time and effort from the team. With good fundraising effort, they can attract and retain the best talent. They offer also huge financial rewards, although that comes at high rate of failure as well as a huge risk. And then for people that stay in academia, which definitely love to learn new stuff, this is also an opportunity to build new skill sets. And these skill sets might not always be useful if, in case of failure, you will be re-entering academia, because there is always a concern about losing continuity of funding. But nonetheless, there's a lot to learn in this new context. So you can see these 2 options as exclusive, but I think it's very helpful to look at this as non-binary question and consider also an opportunity of doing, at least temporarily and for some time, doing both, which can soften and the risk, the transition from one form of activity to another, where a startup can get access to well-equipped research lab that allows for solving more complex issues and digging into the mechanism of action of their technologies. That happens usually through a sponsored research agreement between the startup and the institution. Academic lab in this ways can find additional source of funding within their area of interest, and the founder in this way can retain some academic independence, pursue also other ideas, and maybe become a serial entrepreneur. So this is an attractive opportunity, but it comes with some considerations. And one of them, I think the largest one I can think of, is the fact that the IP in this case is shared between the academic institution and the company. So that comes with negotiations, that comes with additional licensing costs for the startup. There are also some conflict of interest issues that have to be solved between the institution and the startup, that can very seriously limit your ability to take on a leading role in the startup. It really depends on the institution whether that will allow a founder to become, let's say, a member of the board of this new entity. And finally, that will limit some of your academic activities, will impact your time and so on. But it will not limit your need to write grants. So you will need to still continue with this activity. So like in this cartoon from famous Polish cartoonist says, nothing you can't achieve if you really want to, but you do need to be careful what you're wishing for. So let me finish here, and I hope that will open space for some questions. And I wanted to thank a number of people. It takes a village to do translational research from the city of Hope, especially our chemists like Piotr Świderski. There will be two presentations from my lab during OTS. I wanted to mention Damian Kaniowski, who is here with his story on NF-kappa B targeting, as well as the other poster on telomere targeting. Special thanks, of course, to John Rossi, co-founder of the original company of Weistat, as well to Alan Horsager, the CEO of Duet. So thank you very much, and I hope we can talk more later.
Good morning, everyone. My name is Alan Horsager. I'm the president and CEO of Duet Biotherapeutics. I'm co-chairing with Anna, and greatly appreciate everyone here. I think it's going to be a great session. So I'd just like to go straight into it. I'd like to introduce the next speaker. Her name is Dee Datta. She is the co-founder and CEO of Switch Therapeutics, and we'll be speaking about that. But she also has a very interesting background, both in finance, is trained as a scientist, and she'll tell you all about that in the next talk. Dee?
Good morning, everyone. Really excited to tell you about Switch Therapeutics and the formation of Switch Therapeutics, and generally try to address any questions from the audience regarding company formation, especially in the oligo space. So Switch Therapeutics is a next-generation RNAi company. What we have created here is a new class of RNA therapeutics that has 3 strands of RNA, which is a rather unusual structure that you see here. The technology comes from the labs of John Rossi at City of Hope, Bill Goddard at Caltech, and Soumya Das at Harvard. What we've created here is what we call conditionally activated siRNAs, or CASI, and they combine features of single and double-stranded RNA into a single molecule. The 3 strands you see here in this structure, this is one of the topologies. There are different topologies that we have, but they all have 2 domains. One is called the sensor domain, the other one is a payload domain. The sensor domain has a single strand there called the sensor strand, which has single-strandedness and double-strandedness that you see on the left. Having this 3rd strand actually gives us a lot of interesting properties onto the structure. One is that it gives us expanded chemical and structural space that we can put on an siRNA or a microRNA or a small activating RNA. And that allows us to work with different set of ligands and also design in single-strandedness, even if your therapeutic payload may be a double-stranded payload. On top of that, we have payload diversity, where we can put in, like I said, small activating RNA or ASO or siRNA. Most of our work has been done with siRNAs. On top of that, we can design this sensor strand in a way that we can release the payload in all cell types or design it in a way that the payload gets released only cell-specifically. Let me show you a little bit about how this structure actually works. So these structures are very stable, and they get uptaken by cells quite easily. Once they are inside the cell, they get released from this 3-stranded structure through a mechanism by which the sensor strand base pairs with certain RNA markers that are intracellular. The sensor strand is designed to base pair with these RNA markers. If these RNA markers are present in all cell types, then the payload gets released in all cell types. If it is designed to be base pairing with some markers in some specific cells, then the payload will be released in a cell-specific manner. The RNA marker base pairs through the single-stranded region, and then it base pairs through the entire length of the sensor strand. The sensor strand then gets peeled off the three-stranded structure, and the payload gets released. We can apply this technology very broadly. However, we've decided to focus on CNS, our primary area of focus. I'm going to show you some data in CNS. So far we find that this three-strandedness actually has a lot of unique properties in terms of broad distribution, uptake, and potency in the CNS. What I'm showing here is that Casis actually distribute really well in all brain regions. This is a picture of the mouse brain after an ICV administration, and we can see that labeled Casi goes everywhere in the brain. In addition, if you look at the picture on the right side, it shows that they are taken really well into the cells. The clear pink dots show that they are getting concentrated in the endosomes. Following that, we also looked at the activity of Casi, and what we find is that they are very potent and have durable knockdown in all brain regions. What we're showing here is the data from 14, 30, and 90 days after a single injection, and what you're looking at is the knockdown across different brain regions, which are listed at the bottom of the slide. And what you can see is at 14 days, we start seeing very good knockdown. The knockdown deepens at 30 days and kind of stays at the same level even at 90 days. That shows that this class of molecules are very potent and durable. We have advanced this technology in thinking about how do you apply it in the context of cell specificity as well in the CNS. The way we think about cell specificity in CNS is that we try to look at the different contribution of the different cell types in the various CNS diseases. An earlier view of CNS was that everything is very neuron-focused, but in the last couple of decades, it's become obvious that the role of astrocytes, microglia, in various neurodegenerative and other kinds of CNS diseases has been very well understood, and it's becoming more and more clear that all of these different cell types play a key role in disease pathology and disease propagation. So what we have tried to do here is that we've tried to identify the unique markers across these different cell types listed here, such as reactive astrocytes or reactive microglia, oligodendrocytes, and we are starting to build these Casi molecules that are cell-specific. Here's an example of how we utilize our technology. We have our unique bioinformatics platform through which we identify the various markers for these different cell types. Then we pick a marker and design Casis around it. And this is an example showing for astrocytes. We have identified these markers, and then using these markers, we have made these Casi molecules. And here's an example of one of the Casi molecules on the right. And what I'm showing here is that if you have an assay where you just transfect an siRNA, you see a certain dose curve, which is with the black triangles here. And if we put this siRNA in this Casi format for this specific astrocyte marker, and that marker is present in the cells, we find that all of the siRNA is getting released, which is the orange squares curve. However, if that marker is not present, we find that the curve actually shifts to the right, and there is loss of activity. And what we've calculated is that there is a 25x increase in potency in the presence of this marker. We do these sort of assays pretty routinely, and this difference in potency varies, depends on the marker, depends on the siRNA. Here's another example of how we are using our Casi molecule, and this is in the context of a non-CNS application, and this is for cardiovascular. The cardiovascular community has always been interested in finding a solution for cardiac hypertrophy. It is well known that in a failing heart, the size of the cardiomyocytes increase. However, there isn't really a treatment to address that. The cardiovascular community has also known that calcineurin is one of the key modulators of cardiac hypertrophy. However, calcineurin is ubiquitous. It is present not just in cardiomyocytes, but also in immune cells and in the kidney. So there is not a very clear way of generalized knockdown of calcineurin without having any side effects. In fact, cyclosporin, which is a calcineurin blocker, is routinely used for transplantation to cause immune suppression first in patients. So the team also found out that when cardiomyocytes actually go to a hypertrophic state, the level of a particular peptide called ANP, that increases significantly in these cells. In fact, the increase is about 8-fold. And so the team made a CASI with the siRNA targeting calcineurin, but the sensor designed against the mRNA of ANP. And what we did was, we looked at the knockdown of calcineurin in normal cardiomyocytes and hypertrophic cardiomyocytes. And what we found is the CASI is able to knock down calcineurin only in the hypertrophic cardiomyocytes and sparing the normal cardiomyocytes. This is another example of how this technology that was started in academia across 3 different groups has very clear applications in not just one, but multiple therapeutic areas. Finally, I want to talk a little bit about the team. The focus of today's morning session is about building a company based on oligonucleotide therapies. And team is essential for that how we build a team is critical for the success of a startup. A little bit on my background here. So prior to co-founding Switch Therapeutics, I was at an antibody company called Zoma. Prior to that, I was in a company called 47, an immuno-oncology company. And prior to that, I was in venture capital. We have built a team with people from very interesting backgrounds and experience, and that's been critical for the success that we've had till today. Craig Blanchett, who's sitting in the audience here, is our SVP of research. He comes from Genentech. At Genentech, he was leading all of Genentech's internal effort in ASOs, siRNAs, gene therapy, and was leading a large team across various therapeutic areas. Mike Wolf, who is our SVP of Finance and Operations, he was in a large pharma. He was in Amgen, J&J, but a key experience that he brings to us is that he was employee number 1 in a company called Kezar Life Sciences, and from employee number 1, he took the company through multiple rounds of financing all the way to IPO. Siping Han is the lead inventor of the technology and is also co-founder and CTO. And Don and Bob, who are listed here as VP of Biology and Chemistry, bring very deep experience in neuroscience and oligomanufacturing from Roche and also from Amgen. We've surrounded ourselves with very key scientific advisory board members too, again, with all members with very deep experience from the industry. And we are supported by a group of excellent investors, including a number of pharmas. The reason I spent a little bit of time on this slide is that as we go into the panel and talk more about building a company, I just want to reemphasize again that how critical the team is in the early days as you're thinking about the different skill set and gaps, both how you build a team internally, but also how you surround yourselves with the right investors and the right scientific advisory board to really build a company. I'm going to stop there and looking forward to the panel later.
Next, it's my pleasure to introduce Andrew Fraley. He's a serial entrepreneur within the RNA therapeutic space, currently like an entrepreneur in residence at Atlas Ventures and a CTO of a stealth mode company. And he's going to tell us more about like how to build companies actually like within venture firms and from his extensive experience starting biotech. So over to you, Andrew.
Thanks, Anna. Also, thank the organizers for the OTS meeting. I've been coming to the OTS meeting for many years now, and it keeps getting better and better. It's always a highlight of my year each year as I think about what's coming up for conferences. So I thought, Anna and I were talking, I thought I'd give you a little bit of a different talk today as we're in a workshop and we're talking about how you commercialize RNA therapeutics. And I live just outside of Boston. I live in a little town called Arlington, Massachusetts. There's probably even people from Arlington actually in the audience, or a bunch of nerds who all work in Cambridge and commute back and forth. But one thing that's interesting is right by my house is a bike path. And you can get on that bike path and get on your road bike and ride out into the countryside. And you can see the New England country roads and ride all around all of them. And what happens is after you've been doing this for 2 or 3 hours with your buddies, you're on your way back, right? And you're coming back in the city, and what do you see? You see a bunch of kids trying to sell you a cup of lemonade. All right? And I think for many people in the US as children, this is our first thing what we see about thinking about how you start a company, right? It's that first taste of what entrepreneurship is, right? These kids have to go out, find lemons, find water from the hose, get sugar, get, how to make lemonade and try and go out and sell it and get revenue and generate profit, right? And that's what companies do, right? So we talk a lot about what is entrepreneurship. I'm an entrepreneur, you're an entrepreneur. And I think that entrepreneurship is less about a role and a title and a job. It's really a mindset. You can be a PhD student and be an entrepreneur, a postdoc, a scientist at the bench at large pharma. My mom could be an entrepreneur, right? Because I think it's really this mindset. And Howard Stevenson at Harvard Business School really highlighted, I think, what it is. It's the opportunity to pursue resources beyond resources controlled, right? The kid who's getting the lemons doesn't have a box of lemons sitting in his bedroom, right? He has to go out and source that. He has to go out and figure where he can get those resources from. And that's exactly what we do when we start a biotech company. We think about RNA therapeutics. So I thought I'd do for our time today is walk you through how you think about RNA therapeutics, venture model, which uses money from a venture firm to fund the company, and entrepreneurship, and how those things weave together to create a company and drive, commercialize in an RNA therapeutic. And I think there's 3 components which go into that company. The first is science, right? You need the idea, the technology, the therapeutic area you're going to work on, the target, the disease. You need talent. You need people to run the company. You need the scientists. You need the board of directors, the advisory board. And then finally, you need cash, right? You need money to be able to pay all these people and pay for the pipette tips. Okay, so how do you do all these things? I'll walk you through each of these different areas. The first is the science, right? You have to have an idea. What's your idea? Is it a new siRNA modality? Is it a new way to do an edit? Maybe it's a new target or disease you want to apply RNA therapeutics to. And ideas come from multiple sources. I think we all think about academic and pulling ideas out of academic labs, which happens a lot, right? But ideas also come from entrepreneurs who are already working in the field. They come from biotech companies and large pharma and industry who got new ideas they want to spin out and convert into a new company. And finally, they even come from venture firms, which I work with Atlas Venture, where we're looking at ideas that we're interested in and that they want to fund. The reason that drives the value in all this is really intellectual property, right? How do you own an idea? And we do that through patents and trademarks and trade secrets, right? And that really allows us to monetize and capitalize upon all these ideas that we generate. Okay, so how do you put this together from a science perspective? I think the science aspect of building any company, any biotech company, is really about storycraft. How do you tell your story? What story are you going to tell around your science? And that's first your idea. That's your technology. It's that new siRNA molecule you have, right? The target, the disease area. And then the vision. Why are you going to do this, right? What kind of patients you're going to go after? What's the opportunity? What's your unique solution, the unmet need in the marketplace and the clinical world? And finally is the how, all right? That's your mission. What are you going to do? How are you going to pull all these different things together? How are you going to de-risk the technology? How are you going to generate the data and really show you have focus and a team and values that you're going to bring to the company? And so we think about, we're not out selling lemonade, right? Where you generate profit and revenue pretty quickly. We're really following this value creation model, where the data we generate, the patents, the proof of concept data around a particular program, partnerships we do with large pharma, development candidates, and walking that into clinical success really generates a valuation for the company. And that valuation is what we get to tap into after we've made the company successful. Okay, so that's the first section, science. The next component is the team, right? And I think there's 3 pieces to this team that go together to start a biotech company. The first is your founders, right? The idea that they have that they want to drive forward with. Second is your board, your investors. They're going to oversee your company, and they're going to help you figure out how to run it and fund it for you, right? And provide a balance and a check mark and push you harder than you may really want to go. And finally is your operating leaders. Your founders may actually end up being an academic professor who decides not to take an active role in the company, maybe an advisor. And so these are the people that are really going to drive the company. They're going to pull the widgets together that deal with the SAB, deal with the regulatory, the FDA, the legal people, HR department, all that stuff that has to go into a company to really drive a therapeutic to clinical success. Okay, those are the first two components, right? The last one I think is cash, right? You need money to run a company, right? Lemons are cheap. The last time I checked, oligonucleotides synthetically are still pretty expensive. And so we need money to do all this work. And there's two ways we can get cash. The first is non-dilutive, right? And that's where we actually retain equity in our company. That means you don't have to sell your company to somebody else through stocks or options, right? And there's different ways we can get cash for that. Academic grants, government grants, partnerships. You get a check from your dad, right? You can take out money out of your 401k account or your retirement account to pay for your company. The second is dilutive, and that's where you sell equity. And that's where venture firms come in and venture capital comes in. Because biotech companies are expensive. It takes a lot of money to really make a therapeutic drug successful. Last time I looked, it takes about $10 to $12 million just to take an oligo and run that through an IND process, from a preclinical development candidate all the way through an IND. It's expensive. And venture capital is a very efficient manner and a very, I think, smart manner in how you can exercise capital to drive that science forward. And so I'll spend a little time talking about the venture capital model. So a venture fund, what's a venture fund? We all hear about venture capital and all these guys who work in venture capital. It's a bunch of people who come together that build a firm, and they go out to other people and they raise a fund of cash. They get this from pension funds, universities, other large pharmas, anywhere they can find it, right? And they build this pile of cash, this fund, and they have other people that work with them, the support team. They then deploy this cash across their portfolio companies. And I'm an entrepreneur in residence at Atlas Venture, and we assist them by doing diligence on technology and ideas, and then taking them and helping them start those companies and run them from an operational perspective. And either becoming an operating leader in the company, a foundership, sometimes taking board seats as well. And so, how does the venture firm deploy this cash over an idea? It's this notion of using what we call a seed stage company. We want to de-risk the idea, right? It's a small team, and early venture money is there to show, can we take that academic idea and recapitulate it? Can we show it works? Can we understand where we're going to apply it in the clinic? What patient populations? What's the market look like? And then over time, the venture firm continues to add more cash into a Series A. We have a major inflection point, follow-on financing, and finally what they're really interested in is that exit, IPO, a merger, where they can actually now take that value creation and capitalize upon it and have revenue. So what you do is, in our final time, is walk you through a case study, Coro Bio, which I started 4 or 5 years ago with Atlas Venture. Josh Rosenthal, a professor at the University of Chicago at the MBL in Woods Hole, Massachusetts. So our science, we're focused on RNA editing. It was seeded by Atlas Venture and NEA, 2 venture firms. And we had 4 key technical questions that we wanted to answer during our seed stage where we just had a little bit of cash. The first is, can we direct an oligo to efficiently and selectively edit in a cell, right? The second is, can we take that oligo and use SAR and design sequences around that to drive activity, so we knew we had a dynamic relationship between our chemistry and our biological readouts. The third thing is we wanted to show that we could edit across different species, at least just in cells. Can we connect data in mouse cells to rat to NHP into human to know we had a path forward as we would progress into human? And finally, we just wanted to show we can edit in a mouse, right? And this was this data package that we're going to use to go out and raise our financing on for our Series A, which really allows us to build the company out. In addition to all those technical questions and all the data there that shows it was successful, we had strategic questions were just as important, right? The first is, what's our therapeutic portfolio going to look like? What's our initial indication? There's a lot of risk in building a platform company. It's very expensive. So how can we de-risk that and start with an indication that's straightforward, that we know has a clinical path forward, it's well understood, right? And then how do we build a portfolio around that? What other targets and therapeutic areas are we going to play in? Where can you deliver an oligonucleotide, as an example? The second thing is competitive landscape. What kind of competition are we facing? Who else works in the RNA editing space? Who works in the gene editing space? Is that going to outcompete us at some point? And what really provides our approach to be unique and have really an advantage as we think about the competition? And finally, the third piece, which I think is actually the most important, is how you build corporate culture. Corporate culture and how you build a team and how you run that team is the most important aspect as you think about building a biotech company. We really wanted to build a company that was going to have support from our investors, our board, the corporate environment, and really build a company that we have team success, where we could enable rigorous science and differentiation therapeutics and really be able to retain the talent and the people we wanted to hire. You know, a great scientific idea can get easily ruined by a bunch of bad scientists, right? And so luckily, we put all those things together, we put the data together, and we ended up building out a Series A where we raised $91.5 million for Quora develop our Opera therapeutic platform. So where's Quora today? They've raised over $200 million to date through 2 rounds of financing, plus the seed round. They're raising over another $100 million in the coming months as they're going through a reverse merger to become a public company for an exit for the initial investors and the founders and the team members who own stock in the company. They have about 85, I just heard 90 employees. It keeps growing on a daily basis. They have a wholly owned pipeline, so all the therapeutics they work on are fully owned by the company. And so if you're interested in Quora, its technology, happy to chat about that, but I'll also point out Shannon, who's going to be speaking on Tuesday around some of the data science work that we're doing and how we understand oligonucleotide design from a machine learning perspective. So finally, I'll end with one slide and a bit of data for you, just around venture funding and biotech landscape. So the markets and the interest rates and inflation have been going crazy in the past 2 years, right? We've all seen it. There's been lots of layoffs in biotech, and it's been really challenging. I think one thing that's very interesting is on this graph is you'll note in the bars here show the amount of dollars that have been deployed, and the line shows the amount of dollars that have been raised into a venture fund for the past number of quarters or the past few years. And you notice there's a difference for Q1 and 2 for 2023, right? There's a lot of cash and equity that venture firms have raised that have yet to be deployed. So there's money sitting on the sidelines, waiting for the environment to be right for them to start putting this into new companies. So I think that, you know, RNA therapeutics is obviously a growing field. We continue to find great scientific success, and there's cash there waiting for us once new ideas arise. So finally, thank you all, and look forward to the discussion we have in a few minutes.
Lovely talk, Andrew, lovely talk. So what Ann and I tried to do with this session is to really create a theme, starting at the very beginning and going towards the end, right? Starting with an academic idea, and then moving it into a company, and how you get that financed, and then ultimately, how do you position it to other stakeholders, like biopharma? So the next and last speaker is Rabia Ahmad. She is a business development director at GSK. Before that, she did business development at GE Healthcare and is trained as a biochemist. So she's going to tell us a little bit about GSK and what they've been looking at. She also was involved in the deal with Elsie Biotechnologies, which some of you are probably familiar with. So hopefully we'll hear about that too.
So thank you for the introduction and also for the opportunity to be part of the session. I'd like to spend a little bit of time talking about pharma partnering and what that can look like. In the time that I've got today, I'll talk about GSK as a company, what we're trying to achieve and why partnering is important to us. And then I'll also talk a little bit about the common models that pharma use to partner with biotechs, and finish by talking about the deal process and outlining that. So this is just the usual safe harbor statement. We at GSK, we are uniting science, technology, and talent to get ahead of disease. And this is our purpose. And since becoming a standalone biopharma company last year, we have set ambitious targets to be a force for change in the ecosystem. And we are focused on 4 key therapeutic areas, specifically in diseases where there is a patient need for novel therapeutic approaches. Our R&D strategy leverages our expertise across vaccines and specialty medicines to actively seek out new and differentiated opportunities in diseases with high patient need aligned to the 4 key areas that I've highlighted on the slide. So infectious diseases where we are an industry leader with 2/3 of our pipeline focused in this area, currently driving new medicines across challenging diseases like RSV, Hep B, and more. HIV, seeking opportunities to further evolve the treatment landscape through longer-acting prevention and treatment regimens. Immunology and respiratory, expanding on our leadership in key areas such as eosinophilic-driven respiratory disease and lupus, and also investigating potential solutions for challenging neuroconditions like dementia and Alzheimer's. And oncology, which is an emerging area for us, and we've got 3 key research focuses: immuno-oncology, synthetic lethality, and tumor cell targeting. And our R&D capital allocation is focused on these 4 therapeutic areas, and we are focused on developing our late-stage pipeline, bringing medicines to patients as quickly as possible, organically, and also through business development. So we're focused on the novel medicine, but also on data and platform technologies to support accelerated pipeline growth. And we're always searching for new applications of advanced technology to align with our R&D strategy and complement our early-stage portfolio. So we're leveraging new and existing platform technologies to identify and accelerate clinical opportunities. And we have a broad set of platform technologies, and we'll continue to add to this through partnerships and augmenting our internal capabilities. So, for example, through strategic business development, we acquired Affinivax, and also our collaboration with Wave Life Sciences leverages our expertise in oligonucleotides. In data technology, we have access to a rich and large data set thanks to our data-focused collaborations, including a recent agreement with Tempus, which provides access to one of the world's largest sources of de-identified patient data to accelerate drug discovery. So as I said, business development is an important component of our R&D strategy and supports the growth of our pipeline, and that is our #1 capital allocation priority. In addition to advancing our pipeline organically using the science of the immune system, human genetics, and advanced technologies, business development aims to create additional value for GSK, our partners, shareholders, and patients. And here I've outlined recent deals from across our portfolio. At GSK, we're excited to work across the industry as a partner of choice, focused on 4 differentiators that we believe are important for potential partners when they're thinking about a collaboration. So firstly, therapeutic area expertise and leadership. We're the industry leader in infectious diseases, including immunology and respiratory. Secondly, development, scalability, acceleration, and life cycle value generation. We have the ability to develop and deliver complex global development programs at pace. And thirdly, our access to advanced tech capability and platforms and agile deal making. So that's decision-making with agility to remain competitive and create immediate value. And all of this is underpinned by a BD function that is externally connected to source a proprietary deal flow, engaged with the broader ecosystem of biotech, academia, and VCs, and ultimately be ambitious for patients. And we know that by doing all 4 of these things really well, we can be a partner of choice for any organization and feel strongly in our capability as a focused biopharma company to create value for a partner. In terms of the different models used on deals, there are a few, but the exact structure of a partnership will be different for each deal, and that will be determined as part of the deal process in discussion with the partner. So we could use an R&D collaboration, and this is when 2 or more companies come together and work to answer a specific question or a set of questions that could be related to basic research or to a specific product in development. There are strategic alliances, and these are when parties work together to move a product through the development process. So they may collaborate to take an asset from preclinical to clinical development or expand into new markets. Or it could be that the partnership gives one party access to capability that it wouldn't otherwise have access to. License agreements can be standalone, or they can be a component of an R&D collaboration or a strategic alliance. And this is when a biotech gives a pharma company a license to use the product. So it could be that they get certain rights to use, manufacture, and sell a product or technology, and that license could be granted on an exclusive or a non-exclusive basis. It could be limited to a particular use case or to a geography. A joint venture is a new business entity that's formed when 2 or more companies come together and each contributes cash or assets or IP to the new venture, and that new entity will be responsible for the development of the asset. Equity investments are when a pharma company invests in a biotech for a share of ownership of that company, and pharma will often make equity investments while making or as part of one of the other deals mentioned here. Acquisitions occur when a pharma company takes full ownership of a biotech. So they acquire all of the products, the IP, the property, and the people. And this type of deal happens less often, but it does allow pharma companies to quickly enter and establish themselves in a particular field they haven't played in before, or access innovation that might be happening externally. So here I've outlined how GSK has used these models to build our oligo pipeline. In 2019, we initiated a collaborative development and licensing agreement with Ionis that led to the discovery of bepirovirsen for HBV. The collaboration included an option for GSK to exclusively license molecules developed under the agreement, which we exercised, and bepirovirsen is now in phase 3 development. We then licensed an asset from Arrowhead in 2021, and at that time, this was a phase 1/2. It was in phase 1/2, and now that's in phase 2B. And GSK has the exclusive rights to develop and commercialize that asset in all territories except Greater China. And then in 2022, we entered a strategic collaboration with Wave Life Sciences, and that was centered around their alpha-1 antitrypsin molecule, which is a preclinical RNA editing program that's soon to enter the clinic. But through the collaboration, GSK also has access to their PRISM platform for developing oligos for RNA editing, splicing, and silencing for up to 8 preclinical programs. And then earlier this year, we entered into a research collaboration agreement with LC Biotechnologies to explore the value of their discovery platform and P5 chemistry for making novel oligonucleotides. And after an initial research phase, GSK has the option to non-exclusively license the technology. And finally, I want to give an overview of the partnering process. So just as the deal structure is unique for each deal, the partnering process will run differently depending on how many different, depending on many different factors, from the type of the product, the stage of the development, and BD priorities for each organization. And here I've outlined the general steps in a process. So after the initial contact, which could be made by pharma actively scouting for partners, and many companies will have dedicated groups that seek out products that fit with the specific criteria of what they're looking for. But also, the initial contact could be made from the biotech, who also will be looking for partnering opportunities, and we may receive incoming opportunities from biotech directly. There may be an opportunity to meet at conferences or other networking events, and sometimes introductions are made through a mutual contact. Usually, the pharma company will request a non-confidential deck which outlines the company products, what unmet need is being addressed, and we'll use this to do a preliminary assessment of whether the product fits our strategy and that the scientific data presented is robust, it's reliable, and is of a good quality. Some of those discussions may be done under a CDA and utilize data rooms to share documents and data. And at this stage, a good strategic fit and justification for capital investment are key. If we do decide to partner, we'll start to discuss what a potential partnership will look like and put together a term sheet, which is usually a non-binding agreement outlining the non-financial and also the financial terms of the partnership. And once the parties are aligned on that, we'll enter into a deeper round of diligence where we might look at more scientific data, including source data. We'll look at any IP, regulatory, CMC, and manufacturing considerations are also taken into account here, and we'll also conduct a financial and legal diligence on the company. And as long as nothing untoward is discovered, the parties then move to contracting, where a final agreement is put together, and that outlines all of the terms and that will govern the relationship, and also what activities are being done by each party. So during the diligence process, it's important that the biotech is able to address what the product is, how the product works, what's novel about it, what's the data that you've generated, and what are you looking for from a partnership. So with that, I'd like to end the talk for today, and I look forward to the Q&A session later.
Thank you, Rabia. So now we're going to move on to the very interesting part of it, which is the Q&A component of the panel. How many people here are involved in commercialization or are interested in commercializing technologies beyond being a scientist? Okay, a good chunk, a good chunk. How many people are interested in starting companies that haven't before? Okay, yeah, there's a few. That's good, good. So again, what we've tried to do is really focus on the entire maturation of company development, from the idea all the way out to the point at which you'd create something that's interesting to pharma. So we're gonna bring out the panelists and discuss, but I think what we want to talk about really is understanding. At what point, if you're in an academic institution, at what point does it make sense to start a company? How do you finance it? And how do you make something interesting that will be interesting to the people that are going to finance it and the companies that will ultimately buy it? So we're going to do two ways on Q&A. We're going to do people, if you're interested in the audience, you come up to the mics. There's mics at each of the rows. So come out to the mic if you have questions. And we'll also be doing questions from the online community as well. So don't be shy. Is there anyone that has questions? If not, we'll start off. Okay. So I think I'll start by with the first question. And maybe this is a little bit more for Marcin and Dee. Actually, everybody. At what point does it make sense to, if you're in an academic institution, you're in an academic lab, you've done some interesting work, and you think this could be useful, right? At what point do you know, oh, it's time to start commercializing it? What are the cues, or who are the people that you would go and talk to find out if it's the right time to start a company? Because I think timing matters, and I know that there are factors that can influence it, that can make that a dynamic decision. I don't think it's always very straightforward, but there's lots of factors that can influence it. But how do you go about making that decision?
Okay, so I think from academic point of view, I mean, it's definitely the first kind of inflection point for us is this big publication coming out, because that points to the impact, which is usually the timing of the story. It usually doesn't follow an existing trend for a long time. So it's something new that allowed you to kind of see a story published in a high-impact journal. Of course, there is a lot of politics involved and luck in getting a publication out. But being outside in venues like OTS as well can reconfirm that the idea is really flying. And I think another feedback is also from these offices of technology and licensing and a kind of intramural feedback on interest that they get when they start putting up your portfolio for external investors and sponsors to look at. But don't count on this technology office to really do the footwork and really find the best partners for you. So unfortunately, that's not that simple.
I'll add to that. I would say novel technology doesn't always mean a company or novel product. I think it's important to think about if there's really good cutting-edge technology that you've developed, that there's a way to apply it to a specific problem. And I would say the technology transfer offices are a good place to start, but it's usually also good to kind of pair that with some conversations with others in the industry to start thinking about how that technology can really be taken forward to creating a company. And a company will be created if it is really solving a problem, if it is really addressing an unmet need. And then in that process, one needs to think about what are the risks to getting to that point. So sometimes the technology may be very cutting edge and may be very novel, but it still has too many risks before you can put it into a company. So it's important to think about what those risks are. Some of those risks might be possible to address within an academic setting, and some may not be. It may not be possible to do it in academia, and then that is a good trigger to try to raise some capital around it and try to address it maybe in a small way. Like Andrew was saying earlier, that you could do a seed financing as one of the sources, and then address some question, and then take it to the next step.
Yeah, I would also say that from my perspective, having relationships with people who are in academia, who are colleagues of mine who might be at and are in the field, and just having relationships over time as they work on things, and the constant conversations that we might have on a regular basis around, I have this new idea, I'm working on it. Do you see application here? How would you think about moving forward? What experiments in my laboratory could I run that would help convince you that this is interesting and there's unmet need and I can apply this idea to something? What is that data package going to look like? Because I think that can help, as you would be running a research and academic lab, think about what's the data you need to generate for someone to really kind of take notice of it. So I think that relationship and those conversations with people in industry or people at venture firms or wherever they are, angel investors, that over time that evolves, I think, can help you figure out when is the right point to move it over and if it's worth moving over or not, right?
So this is sort of a leading kind of comment on that, or question. But do you think it's appropriate for academic scientists to form relationships relatively early with venture firms, even if they think themselves, well, it's probably not ready for commercialization, but I want to know when that point is, and what do I need to do to get it to that point? Should they start talking to folks at venture firms even that early?
Yeah, I think the earlier you talk to them, the better, in fact, actually. It's obviously that's what you're interested in doing, right? If you're interested in doing lab work, then,
That's the assumption.
That's the assumption. But I think that's important because that helps you figure out, what are the key experiments I need to run? How do I really think about moving this forward? Do I want to move it forward the way they're suggesting? And every venture firm and every investor and every biotech and pharma even has a different viewpoint on what that's going to look like, right? So you're going to have to triangulate across a number of different people and teams and groups to kind of understand that. But I think the relationship piece is very important. In fact, I've seen a number of professors. David Liu's a great example as a keynote speaker. I worked at David's first company called Ensemble Therapeutics, and he built a relationship with, with different venture firms over time. He has one with Atlas. And every idea that's come out of his lab, he's had that continued relationship with a venture firm to say, hey, I got another idea. Do you think I can move this forward? How would I move it forward? What do we kind of resource do we need to put on it? Would you be interested in it? And they've already been primed up, right? So as he starts to think about that, and as universities think about how you file IP around those ideas, you've got people in industry who can help you kind of navigate some of that, which I think is really important.
Yeah, sure. I was actually thinking that it might be interesting to dive a bit deeper into this guidance that you might be sort of getting from both the investor community, but also from the pharma business development people, right? Because we know that interest fluctuates. We know that the oligotherapeutics field now is at a high, but it's a very dynamic environment that people want to see different things depending on who is leading at that specific time the strategy of a biotech company or pharma company, or I guess like what partners in a venture firm are interested in. So I'm just curious to like understand like how you guys think about like navigating these changing type of questions that you're going to be answered and expectations that your companies are going to have to face over time.
Try that. I think with changing times, sometimes you find some therapeutic areas or modalities really hot and in favor, and some are not. It's not that they're completely ever out of favor. I think the bar changes. Sometimes, the bar for the entry point for starting a company, if it is in a very hot area, that lowers, but if the bar will increase if for some reason that's out of favor. I mean, we are seeing right now the whole AI field. There's new companies starting almost every day, and that's hot, and that's. But I think right now the bar, the entry point is actually much lower right now, and that's just something to consider. The other thing I would say is to understand what is the common risk that people are trying to address for a particular area. For example, I'm coming from a proteins background, so my training has been in protein engineering. I've always worked in protein therapeutics company, and when I was starting Switch Therapeutics, one of the common theme, doesn't matter whether it's our technology or any other oligonucleotide technology, the common theme, the common risk was delivery. It didn't matter what you're doing, which organ you're targeting or which target you're going after. The first question is, can you deliver? Can you deliver in sufficient quantities without any tox effects to the target organ, right? So it's very important to try to address those very early on, irrespective of what the investor sentiment or pharma sentiment is, because those are some common risks, and it's good to try to identify those early and get those addressed sooner in the process.
I can add to that from a pharma perspective. So, as the question asks, strategy does change all the time. But the thing is, we get like hundreds of opportunities to evaluate every year, but we don't and we can't do hundreds of deals every year. So sometimes the answer is no to a particular opportunity that comes in, but that doesn't mean that we don't think that that's in exciting science or that it's off strategy. It could be on strategy, but we have a certain amount of money to invest also, and we have to make choices, and we have to prioritize where we're going to allocate that capital. And so sometimes the answer is no, but what's important, to Andrew's point, is to maintain that relationship that you're building with the pharma company, stay in touch with them, provide regular updates. And as things change and evolve in a pharma company, it's really important to have got that relationship already established so that it's easy to reach back out to them, or for them to come, reach back out to us, and for us to explore an opportunity to collaborate in the future.
So Robbie, on that, just to follow up on that, and it's going to go back to our first question. So do you, as a pharma company, think about technologies? When you look at technologies, do you prefer to see some level of commercialization already done, or do you actually, I know that there are collaborations that happen with academic labs, but from your purview, do you actually think about relationships with academic scientists as well? I certainly understand from a venture perspective how that works, right? You work with the academic scientist, you may even found the company with the academic scientist, but from a pharma perspective, is that just too early, or is there good value? Because I think one theme here is keep the end in mind, right? Where is this product going to end up? And so talking to someone like you could be very valuable for an academic scientist as well.
Yeah, no, absolutely. I think we're focused on early stage and late stage opportunities, and early stage could be very early, so it could be something that is, still very research focused. At GSK, we follow the science, and all of our disease targets or indications have a very strong scientific rationale. But what that means is that we have to invest and also support groups who are doing the science to understand the biology of disease, to understand how the therapies are working. And so, collaborations in the early space are definitely, absolutely on strategy and things that we're actively seeking and scouting for.
I think we have the first question from the audience. Go ahead, Puneet.
Okay, is my yeah, it is. All right, so my question had to do with conflict, right? So as a academic founder, right, you have a certain vision for what you want to do, and then your venture partner wants to do something different. So when, I'm sure that happens quite a bit. So how do you guys resolve that when you get to that situation?
It's a good question. I think it highlights the need for these early interactions, because it comes in a discussion basically between the academic founders and during post-pitch discussions, what potential interests from the other side can be. You might be presenting something which has very specific application. You think about big therapeutic. They might be more interested in just an aspect of delivery of the whole technology. And that, requires some kind of rethinking of the idea. And as an academic founder, you might not want to move along this path, or you might agree that this is actually something that can build up in a much bigger story than originally thought. So I think this highlights this need for early on interactions with investors, companies, and so on, to basically revisit your original intentions and so on for the original finding, which we come up with a very well defined idea where to go. But this is usually a result of a kind of a research process, a scientific idea, which involves completely different set of kind of completely different thinking process than what you apply to commercialization, which might just pick an aspect of the whole story. So definitely, I think it's a going back and forth process of interaction with the other partner.
Yeah, I would say, I think also too, things maybe are a little messy in the early days from an academic lab that you're transferring something in or working on. But eventually, over time, the academic lab and the company are going to start to have different focuses, right? Academic lab is typically going to want to take their ideas and continue to run with them and go down some rabbit hole that they have just curiosity around. While the company is obviously more focused on how do I go from point A to point B to start to make a product, right? And really have goals along the line of that. And so I think what you see typically is you start to see just interests start to diverge a little bit. And the company then starts to think about other resources it can bring on outside of just that maybe initial idea, wherever it came from, that'll start to bring in, other companies or other aspects of their technologies that it's going to need to make the place successful. But I think as those run, one model I've seen works quite well is thinking about having supported research agreements with a founding lab or another academic lab that you might just identify because the company needs their support and their know-how, where you can fund some basic research or some data that might be very beneficial to the company, but at the same time is not necessarily always creating intellectual property that the university may own. The company can really wants to own that. And there's ways that you can kind of get both at the same time. Academic freedom, publications, and then also IP and, idea to product, right?
And I think I wouldn't underestimate the active effort that you can put into actually bringing people on board that are completely aligned with what the end goal, and what the mission for the company would be. And then once you have that alignment, you might have disagreements on how to get there, but you're actually clear on we're all driving towards the same direction, which I think generally makes that conflict a lot easier to deal with.
Thank you. Yeah, I just want to build on that a little bit. So say you're an academic lab who's at that point where you've decided it's time to partner and there are multiple people interested. What are some red flags we would look for in who the wrong partner is? And from your side, from the pharma side, what are red flags you would look for in an academic lab when making those partnerships? Are there any tangible things that jump out?
Yeah, I think from a pharma perspective, so what makes an opportunity particularly attractive to us is one where there is a real benefit to patients as a result of that. So here I'm talking about when a company has an asset or they have a product that they want to partner, not just sort of early basic research that needs to be funded, but where a company has a product. If the company really understands what is the impact to patients from our technology, or what is the improvement to the pharma R&D process from this product, and they're able to articulate that, that's impressive. But also to understand what are the differentiators of their product, and also what are the risks, and have a clear strategy for being able to mitigate some of those risks. That's what makes a company particularly attractive. And when companies don't have that, then that's usually a sign that they need to go and start to think about whether they're ready to partner.
I just might add from an investor standpoint is that biotech is slow. It takes time and a lot of resources. So as you talk to potential investors, they really need to share the vision of where you're going, because this is not a quick thing that happens, right? It's not an app, so it can take quite a bit of time. And so I think aligning with those investors to make sure that they understand how long this is going to take, and each of the steps as well. Like what is it going to take to get from here to an IND filing, and how long is a phase 1, and the capital requirements associated with that. So as long as you can align on those, I think that'll help you identify the right partner.
Yeah, I'll just add to that. That's actually critical. The alignment is critical, and along with alignment is trust. Is that if you're going to get into a partnership, whether it's a pharma or whether it's a VC fund or whether it's other team members you're bringing in to build this, you are in for a very long-term relationship with any of these parties. So building that trust very early on and having that relationship that that this is not going to be a smooth ride. Any startup is not a smooth ride. It's going to be rocky. There are going to be ups and downs. So having that trust and that relationship is very critical to build that.
And from founder perspective, I think one of the red flags could also be, I mean, you want that what becomes the asset, your technology, will be valued critical for the investor as well, for the company. You don't want to end up as one additional asset that kind of fits into their portfolio, but might not be pursued in the long run. So that's a concern. It's hard to guess, but yeah, you want a high value and kind of focus on your technology from the other side.
Thank you.
Alex Garanto from the Radboud UMC. So I have 2 questions, but before asking them, this is an educational session. So young people, if you have questions, come to the microphone. Don't be shy. Shy. Yeah. So you can come. So I have 2 questions from a naive academic researcher. So you have been talking about investors, and well, you have to find them, but what do they get in return? Because I don't know, I don't have any experience, so they get part of the company or whatsoever. And then you also have been talking about successful stories, but does it mean that if your startup fails, it's the end of an entrepreneur, or any of you have any experience with failing and then having a big success in another company?
Yeah, I can say that failure is par for the course, right? We fail all the time. Venture companies probably in their portfolio, I would probably say at least 75% fail. They're not all successful. And the ones that are super successful are very, very rare. If you look at the average return for venture capital in companies, it's usually 3 to 5x of their investment. It's not a multi-billion-dollar company. Those are very, very rare and very much gems. But I think personally, as you think about companies and their success and failure, right? An idea in one idea in one company that you might pull out, it could go to one company, and you could get a lot of work done, but it might fail because you don't have a lot of cash on it. I started a company called Triplet Therapeutics that was focused on using ASOs for triplet expansion diseases. And at the time, we built a small team of people. We raised a bunch of cash on it, and we got caught up in that the data we generated from our GLP study, we had some neuronal cell death. And at the time, Wave and Ionis were also working in Huntington's, and they were struggling with their stuff in clinical trials. And the market decided that, and the investors decided that this was an area that they felt was too much risk and they didn't want to play in, right? And so Triplet actually closed, right? But that target, MSH2, is still being used in other companies. The IP is still there. It got moved over to CHDI, and people are working on it. So just because a company's a failure, a startup's a failure that you might have had out of your lab, that can be reincarnated into new entities over time if it needs to be. It can be recapitalized and used again.
Yeah, I've had failures as well. So I was a research assistant professor at USC. I was working on a technology. It was optogenetics for folks that are aware. I'm a vision scientist by training. I thought this could be a great approach for treating blindness. Huge amount of risk. This started in 2007. Capital markets imploded around 2008. So we were out trying to raise venture capital. That wasn't happening. We had one shot on goal to try to get it translated into larger animals, and it was tricky. It was very tricky. There was a bunch of things that we were doing, a bunch of things that I learned there, but I would say it's painful when it happens. It's painful as an entrepreneur. But you can either, go lick your wounds and then get back out there and do it again, or you can, say, that's not for me. So I encourage people not to be discouraged by failure, because it's rampant in this. This is high-risk stuff, but as long as you accept that, and you can't take this stuff personally. You really can't, really can't. There's so many ways in which things can go wrong that are beyond your control.
I think there's I guess, cultural, quite large differences between, how Europeans deal with failure versus Americans, right? And this is one of the things that I definitely think that we need to learn a lot from, our American counterparts, because you can't let, that fear of this might not work out be something that actually stops you on that journey. And in general, when talking to investors in America, they don't see you having a failed previous company as something that is a barrier for them not to invest in your next one. They see it as you've now made a substantial amount of learnings that you can then apply onto your next opportunity. So I think, there's, as long as you learn from that step, it's totally worth it.
Hi, too close. Hi, Phil McGilvery, HC Biosciences from Boston. I wanted to ask, as someone who's done a lot of pitching for seed rounds in the past, and who imagines there's a lot of people in the audience who are hoping to do seed round pitchings in the future, what are the key points for a productive conversation with investors of any kind when seeking funding? Thank you.
What are the key points? I think having a clear, crisp story, right? That you know your space, you know the what, the why, and the how, right? And you've worked that out, and you can clearly state that and show what that means, and show that you understand the differential and where you'd like to take it, right? But I think, and also being open to the fact that that's going to change as you start to work with an investor, because they're going to have their own ideas on how to drive that forward from their experiences as well. So, and I think it's also the relationship, right? So I think that idea and that story matures. So when I've gone out to pitch companies and build companies, a lot of times I don't take it to the first venture firm that I really want to get the money for. I take it to somebody I think is more junior, right? Or might be a smaller firm, who are someone I don't want, because I want their feedback. Right? So I'll take and pitch the company and pitch that story and really have it clear as possible, and take their feedback and solicit that, and use it to turn the crank on the idea one more time, mature it, right? And have, the components, those 3 components I was talked about, who's the team, who are the people who are going to back you scientifically, that you've got advisors around the table. They want to see that you've got smart people who are going to work on it. And I think it's that maturation process and that relationship build. You might go back to the same VC 3 times before it gets funded as you mature your idea. So I think knowing that it's a process and it's not a one-off is where success comes.
I'll just add to that when you're doing a seed round of financing very early, you don't have a lot of data, right? So you have a vision that you're trying to sell. You have some high quality science, but not a lot of data. So to build a team around that is actually pretty critical. So to build a team around who believe in that vision. So whether it's advisors, potential scientific advisory board members, but also if there are members who have committed to working on that idea and who are coming with very credible backgrounds, I think that goes a long way for financing.
I would add one more thing. I think that risk, no one likes risk, right? There's a lot of failure. And the more you can take risk off the table for an investor or just for the company scientifically, the better off you're going to be. And really showing how you've thought about risk mitigation, right? What's the cash going to provide you to get to a go, no-go decision? How are you going to say, I'm going to take this money and I'm going to get to here to tell you if I can run it or not from a seed perspective? And really having real clarity on that, I think, is really what helps when you don't have the data quite yet. Really figuring out how you de-risk an idea is very important.
Can I just add one last thing? I think of the pitch deck as sort of a set of risks that you address. You may not know all the answers, but you know how to think about it. And I think what I would say is that it's really important to develop a relationship with someone in the venture firm and have an ongoing dialogue. So yes, I wouldn't think about pitching as transactional. I would think about it as the first conversation in a new relationship.
This is a great panel. I want to congratulate the organizers for making this panel. And we are all learning from this. I have some science questions, but I won't ask them first. But something related to the theme. First, I will ask Andrew Fraley the question. Andy, you pointed out the graph that how much money is sitting there. And at the same time, we also see some disturbing news of people getting laid off and the kind of a slowing down recently. So it's a kind of a hot and cold together. Considering all the young people who are entering the field, what is the momentum? What needs to be done in a focused way to utilize the money and change the direction? So that's question for you. And I have another question for Marcin.
Yeah, that's a great question. So obviously we've had, over the past few years from COVID and before that, just, a massive slush fund and tons of money that was pretty frothy for biotech and investing. And I think that it created a ton of companies, and people are throwing money very much very at risk, right? And you saw a lot of companies getting started, and some of them probably shouldn't have got it started, to believe with. I think that you can have too much money floating around, if you will, right, from an investment perspective, because the right amount of cash really just drives right decisions. And so where we are today, I think that because you've seen the markets and interest rates go up, and you're starting to see this challenge that the markets have really kind of moved, to general markets, not the biotech investors, have moved away from investing generally in things like the MBI and in biotech generally, where you've got clinical programs and you've got things that are really trying to get into become an approved drug. And so a lot of these companies have been out there and have done a number of rounds of funding when the money was frothy, and now they're getting to the point where they need more cash. They need cash from the public markets, and they can't get it right now because the stock market just can't provide that due to interest rates and just a general decision that biotech's a little cool for them right now. And so they're getting to the point where they've got phase 2 data, they don't have a lot of money in their bank accounts, and they can't get more cash. And so you're seeing a lot of companies that are struggling with data around a phase 2, phase 1 program or IND, where the markets are looking for the next thing, or they're looking for the next thing to move the company forward, and the markets and the cash and the company aren't there. So you're seeing some of these companies start to close, I think. And you're starting to see, as we point out, that's been tough on the layoff side. But even still that all going on, I mean, I think when I started working, I had to go interview 5 places. Man, I'll even interview with you, right? And 2 or 3 years ago, we would hire our associate scientist within 24 hours of interviewing them because you couldn't find anybody, right? So I think that while we're seeing some of that right now, I still think that the environment's still strong for positions. And so I think from that investor perspective, what you see, the money that's sitting there, that money is still sitting on the sidelines, right? Because I think the venture groups are recognizing what's going on in the markets right now as later stage companies can struggle with getting access to bank cash and cash you might see from in the public markets, and so they need to support their companies they've started. It's that long-term relationship. You want an investor who's going to sit there with your company for 5, 10 years and continue to support you. And so that's why I think that company hasn't been deployed yet. And as as inflation gets under control, we get interest rates down, hopefully, you'll start to see that cash start to come into new companies.
Great answer. I'm glad you didn't come to Alnylam now in a position to give money to Alnylam. Good. So now the question to Marcin, if I may ask, this is regarding the academy-industry relationship. Let's say you have a postdoc who has a new idea for Duet, and he wants to publish that paper, he or she wants to publish it in Nature, and ready with the data and so on. And whereas that Alan says, this is a great idea, don't publish it, and the others will take it and find another company and so on. How do you deal with that conflict in an academy industry perspective?
It's a great question. That's I think that's part of having a team and trying to also kind of nurture and mentor people. So I think it is a very, very likely scenario. Something big comes out, but you have your obligations. That's I think part of this conflict of interest scenarios that you have to consider. So lab members, of course, have to be aware of your obligations and interactions with companies that you founded. That has to be clear. And I think institutions now regulate it quite clearly. So this is one of the initial pieces of information that any new lab member needs to get. So they are familiar of your of my obligations and or involvements. So I think this is the moment where you have to you can't constrain your basically mentees because you want to see them succeed, you want to see them driven. If you start deflate them early on, it's not a good idea. But at the same time, I think they also change mind when they get more involved and also more familiar with what is being done on the side of collaborating with a company. I think when they are involved in this sponsored research agreement, they also see what type of research is being done with the company, what can be done in the lab. So they might be able to develop their own mind where they fit better. So whether they want to pursue this idea, which might get narrowed down to some technology, but they want to see it moving forward and maybe becoming a drug and having this bigger impact, the ultimate validation of getting to the clinic, or they're more interested in developing it into kind of a research tool and maybe using it just to dig deeper into some biological mechanism that's really their favorite. And so I think that's kind of an ongoing conversation that has to be part of the lab, and that will allow kind of both sides to settle on some. There's always a compromise that can be reached.
May I respond as well? I think we are actually, just very specifically, we're very interested in Marcin publishing and in people from his lab publishing. Because that's good for us, right? That's a milestone. Assuming it's the stuff that's related to what we're doing. I want him to publish anyway, right? But particularly when stuff's related to us, there's a process, right? And you know this well, right? You want to, if there's new technology, you want to make sure that you go through the technology transfer office, that IP's been filed, and that rights have been sorted out, and all of that. And that usually you can do in advance of a publication. But we're very pro publication for sure, because we want that information to get out there, and it's a it's a point of pride, and it's a milestone for the relationship. And the other thing I would say too is that. For companies, what we can do if we want to have a little bit more, quote unquote, control over what's going on, we develop formal relationships through SRAs, so scientific research agreements, and that helps us. We don't necessarily direct the research by any means, but we have more ongoing, more involvement and conversations around the science that's going on. So, I think both can be done. Both can certainly be done.
Touching again upon the subject of failure, what is, in your experience, the biggest cause of failure in a project? Is it the basic science behind it? Is it the translatability, the finding the right indication? I'm curious about that.
Well, research-wise, I mean, research is unpredictable, that's for sure. But you always rely on some existing data, findings from other labs and so on. But that's, you can only trust your data at the end of the day. So sometimes, I mean, the failure is basically an inability to reproduce part of the data that have been published somewhere. Doesn't matter how high. So that can be confusing. I mean, the multiple possibilities of why the failure happened. That it can be just inability to technologically advance some project forward. For some of them, there can be complexity in synthesizing some fascinating molecule that you want to do, and or you might not have access to in-house facility that could do it for a reasonable cost. So you might just face economic issue as a research lab, not being able to pay up front, let's say $20,000 just to test a simple idea with external lab synthesizing for you a modified oligo, even at small amount. They won't do it, at small cost because they will need to kind of run development process. And as an academic researcher, you cannot just cover that if your postdoc has an interesting idea, but it can be 9 in 1 chances that it's going to be a failure. So there's a lot of risk that can be just pragmatism taking over. So it does help to, I think, to minimize the risk through collaborations, finding labs that can know the technology better than you or can give you access to know-how. But there are some, I mean, it always helps to make your institution aware of what you're doing and get their support and maybe build up the facility that you would really need to have. And so I don't think that kind of DNA, RNA synthesis facilities are still very common, even in the US, in academic institutions. Now, of course, there are outside providers of oligos, but the cost is still really high, except for the very standard molecules that you would be using.
So your question's about failure, and failure also depends on what stage, right? So is it failure to raise capital for your startup idea? Is it failure after you raise seed financing to get to the next stage? Or we're also talking about failure of, phase 2 companies, which because of the capital markets are not able to raise enough to take it to the next round. So I think the question on failure depends a lot on what stage you're at, and the reasons for failure can be very different based on that stage. So I don't think there's a common theme, but for each stage there may be a common theme. So sometimes you're just not able to raise money straight out of, a university because of various reasons. If you want to raise seed capital, we touched on that a little bit, it has to be a very clear story of why do you need that money, what is the key data point you're going to get with that, are you surrounding yourself with the right team to get to that point, right? At a later stage, the failure could be completely different. It could be the target. Maybe your science is working beautifully, but you just chose the wrong target, right? So I think the question on failure depends a lot on where you are at in the company.
Yeah, I think that we look at failure as something bad. I don't think failure is bad. Failure can be very good, right? Because you want to get this notion of fail fast, right? If I have an idea I want to know technically, scientifically, is it going to work? And I don't want to spend 5 years there and working on it, because if it's not going to work, I want to know fast so I can move on and do something else, right? So there's obviously things like market and technical reasons, scientific reasons why things fail, which are kind of beyond our control, right? I don't want to take a bad compound into the clinic, right? But I think the one main reason why, if you ask about the routes of failure, I think the biggest route to failure I've seen at companies, and companies starting and working, is the team and culture. If you don't have good people around you who are smart scientists, good backers who are patient with you and understand to put the money in and understand the risks, and you don't have a team of people that work together and you can build a culture of rigorous science and have a good place where people enjoy coming to and working together and work well together, no matter what your idea is, it's going to fail.
Long Chen Li from Rocket Therapeutics. My question is on product development for early stage biotech. We know that for oligo development, once you narrow down from hundreds of sequences to your final one, and you can continue optimize the chemistry delivery component. It will take months until you satisfy with the potency and the safety. My question is, how do you balance between developing the best possible compound and how fast you can quickly advance your compound into patient? So is the so-called minimally viable products concept applied to drug development? Thank you.
We have this problem with Marcin because he constantly is producing new stuff. That's very good.
Initial stage of, I think, building up the, coming up with the new approach with translation potential. I think it's all about using proper best available models. So the modification of the molecule itself, there are thousands of ways to modify oligos and so on, and you will hear from different groups, different opinions on how to do it, what else could be done. So you could spend several postdoc lifetimes just running in circles and trying to improve if you don't have a clear-cut models where you can really test and look for a set of minimum goals you want to achieve. So for development of immunotherapeutic drugs, and I think it came up multiple times in presentation by Art Creek, for example, here. It's critical to have readout in the form of responses in human immune system, whether starting in ex vivo setting or later on. Right now we can finally build up some still imperfect but humanized models of cancer, and humanized mice can be used for testing immunotherapeutic drug, which gives you, I think, gets you as close, I think, in terms of testing to reality as possible without having a phase 1 clinical trial. So I think having and building up, spending time on building up the model rather than just spending time on endless modifications of a product without a clear-cut readout, trying just to meet some abstract standards that you know from literature or that have been fed to you by others is critical.
Hi.
I would just add to that is that whenever you're starting on a program specific target, usually you start with something called a target product profile, a TPP. And that gives pretty good guidance in terms of what do you need to see in your molecule to make a decision whether you take it forward or not. Because, if you want to keep making something better and keep improving, there's no end to it. There really is no end to it. The innovation has no end, right? But at the same time, at some point, you need to say that, okay, this is enough. But what is that enough is usually defined by input from various different groups within an organization, all the way from clinical to regulatory to translational group, manufacturing. All of those factors kind of go in to help you determine when is it enough. And once it meets that profile, that's usually a pretty good criteria if you have the right people helping you make that decision.
I would just say that, just as the other panelists have said, that don't spend a lot of time trying to create this perfect molecule. And just don't let that stop you from reaching out to a potential partner. We don't expect, or we are aware that that first generation molecule that we license or we partner on may not work, and it might be the second or the third generation that works. And what's really important is having a really good relationship between the partners, so that there is that trust there that, okay, this molecule hasn't worked, but the next one might, and we're willing to continue the collaboration and invest in the optimization that's required. So don't let it stop you from, don't chase perfection, don't let it stop you from reaching out.
Hi, I'm Vivek from Oligonucleotide Therapeutics. I have a question to Ravina, and then exactly what you just was finishing up. The partnering opportunities, you have shown that's kind of 5-step process. And there's always a race, between money and the time. Everybody has this thing in the mind. So my question is, what kind of a timelines you have? Because this is a very back and forth processes. And as you mentioned that you get hundreds of proposals and you have to narrow down certain ones. So my question is, what is the timelines and how do you really come and get out of these challenges in terms of the both parties, basically those who want to work with you and you also want to work with them?
Yeah, it's a really good question. So there isn't an easy answer because it really depends on the product or the asset and how complicated a potential deal might look like. And that will determine how long the diligence period is. It will determine how long the contracting and negotiation period is. And so it varies on a partnership by partnership basis. I mean, there will be some companies that we are in discussion with for a year, and there will be some companies where we've done deals very, very quickly in the matter of a couple of months. And that again, just down to the complexity of the deal in that specific partnership. But also, sometimes there are, other priorities in a company, and there isn't an opportunity immediately, but we want to continue that discussion. So I appreciate that on the biotech side, there might be a certain urgency to get a deal in place because that's important for the future of the company. What I would suggest is that you're very upfront about, if that exists and you're talking to a pharma company, be very upfront about that and say, look, we really need an answer, by this time because of X, whatever that reason is. And I can tell you if it's a good opportunity, pharma companies don't want to let that slip away, so we will take those into account. And we try and get things through as quickly as possible, but pharma, again, is also, quite slow sometimes, and we have quite lengthy internal governance processes that we also have to take all our deal cases through also. But I think if you're transparent about any time pressures up front as part of negotiations and discussions, that's always helpful.
Hi. As a PhD student, it can be quite hard to imagine how we move from our training experience to maybe sitting on such a panel as yourselves. So do you have any advice from your experience for how to transition from our training experience to such careers that you're in now?
You're already doing the right thing coming to OTS because that's such an interactive space where you can meet a lot of people from industry, a small biotech as well. So that gives you very good exposure to a different world. On the other hand, if within your institution there are projects that move to this translational path, it's good to interact with these groups. It's really hard to get out of the academic world because we have our own kind of space and criteria and so on for success. But it does help to look around. So, later stage, you do want to get familiar with your Office of Technology of Licensing, because they can be really helpful, and also, I mean, they can provide you with a good information about what they are looking for, because for institutions they will be representing you and academic researchers in the institution. They will be creating these portfolios that they can show off to the world. They are not very effective in my experience, but these are very interactive people, good to be familiar. If you start the company, you might see their other face because it will be on the other side of the story then. But having friends in these technology licensing groups is really helpful and a good experience for us, because they usually they provide you with first contacts, and then you can grow your own. Look for also, I mean, there are different trade associations or biotech support groups in every area, even in Southern California, which started to become a growing biotech hub. So I would suggest interacting with them or signing up for some courses, which, for example, we did that, I don't know, 5 or 6 years ago. We signed up for, there was a course in entrepreneurship, which lasted six weeks and so on, but it was a really interesting introduction of what needs to be done and how, giving you also exposure to local entrepreneurs, presenting on how it's being done or what do they expect, the local VC groups and so on. And that kind of puts you on the track not to either invalidate it and say, well, no, that's not something what I want to do, or you get the bug and you turn into somebody who's really interested and want to pursue it. So I think there are ways to enter the field, and that's, I mean, being here is a really great first step.
I would, I mean, as you finish your degree, I think it's like, where do you want, if you're gonna move in industry, what kind of companies and what are you going to apply for? Where are you going to work? You can go to large pharma, right? Are you going to go work at a small biotech where you're going to see more experience and get insight into how a company operates? You'll have more exposure to the C-suite, to the SVPs in the company, how they're running things. If a company of 20, you see a lot more, you see different things than you see at a pharma, right? Not that either one is a bad choice, or just different choices. So I would say, maybe to continue the theme, is figure out how you can put yourself in environments where you get exposure to things you're interested in, right? And that's entrepreneurship. Maybe it's a smaller company. Maybe it's going to business school, or even going and working as an associate at a fund, or going to a consulting group where you get to see lots of different things, right? And you get exposure on those different areas. I would be open to not, just if you're interested in that, just open to not saying, I'm going to spend my life at the bench, right?
Yeah, I'd say that maybe like one of the most wrong assumptions I see people making, like trainees making, is that while being junior, you won't be able to like reach out to key people within the field, or like key people that like you really like look up to, or like key people that like have taken steps in their career that are similar to the steps that you want to take, right? And in reality, I'd say like 80% of the time, you reach out to someone being thoughtful about their time and thoughtful about what you're asking for, right? And sort of like having like a targeted question on something that can make a big difference on how you think about where you want to go next. Most people like are, very happy to actually like engage with you and give you within those 15 minutes learnings that, take you 6 months, 1 year like ahead of your current thought process. So I would say just really take the opportunities of, driving your own learning path by leveraging the people around you. And then again, like this type of meetings are a fantastic opportunity for that.
One last note, as a PhD student and potentially as a postdoc, those are the folks that are creating the innovation. And those are the people that are very well positioned to be the entrepreneurs for companies. So I think I would say that if you're interested in entrepreneurship, that can be a great jumping off point. Now, there's things you need to learn and things people you need to talk to, but I think that, it may be sooner than you think.
Hi, my name is Tiberius Stan, and I'm a coordinator of Validation Lab at LUMC in the Netherlands. This was really a great initiative from DPP to work both and offering both in vitro and in vivo platform for academia and also for the companies to test their compounds using our platform. It's really great initiative, but I wanted to ask you, and also the whole panel, at what point of your career you decided to be like, or even now oscillating between being more like a scientist or more like a business person? And how did you upgrade yourself with the time?
For me, I'm still kind of in between in this intermediate phase without really making full jump to a startup world, and still kind of retaining academic career, because there's a lot of about academic careers in terms of exploring different fields. It can be fascinating. It's of course draining as well. So doing both things it doesn't make it easier. But yeah, I think it does. Depending on the project, and for some project, I had the feeling that technology that we have and the fact of testing it in complex models requires really well-equipped lab. So that was my major consideration, that you cannot really do it with a startup, having a one incubator and PCR equipment and cells on the plate. When you're really thinking about the immune system and its complexity. So primarily that was, I was driven by the fact that you need far more, and kind of academic resources can really help. While at the same time, it could be beneficial for a company as well, driving it faster forward, de-risking in proper setting, proper models, and so on. So this is why I have not made a full jump, but I'm considering still. So it's still active process.
I made the decision right after my PhD to start a company, and that's what I did when I finished my PhD, and there's some technology that I had developed along with my PI, and commercialized that, and that was not successful. So that was my taste of first failure. But then after that, I went to business school, and then I joined a few different venture capital firms. I joined particularly one group called the Column Group, where it was very focused on company formation, so taking technologies from universities, putting teams around it. I learned a lot, and then I went to another company which had 20 employees, and did a lot of business development deals and set up the company for IPO. So I think for me it was more about building up a lot of different experiences after my PhD, including failures, and that's, I think all of that has really helped me build Switch today.
Yeah, I think I'm in the same boat, right? Like working at companies and then moving on, either because you're no longer needed, you've played your role there, and what you're going to do next. And career-wise, my first boss was an entrepreneur. He had started a number of companies, and I got the itch from him a little bit. But I think also, too, realizing what you're good at and not what you're not good at. I realized that once the HR department started in a company, I really didn't like being there anymore. And so I knew that I liked being on small teams. And I enjoyed that early stage of things. There's some people really enjoy late stage pharma. They really want to see a clinical program run or work in a big organization. And I didn't like that. And that was not what I wanted to do. And it took me a while to realize what I was good at. And I'm still figuring that out. But I think that process of failure and reevaluating what you're doing is kind of how you progress, right?
Yeah, so for me, I think it was a personal decision. So I had a close bereavement in the family, and that's what made me realize that, there's a lot of innovation and potential solutions for patients, but how do we get those into the hands of people who could really commercialize them and make them available? And that's what attracted me to a career in business development.
So I think that's an excellent way to end at 10:59, 0 time. So thank you, everybody, for your attention. Anna, did you want to add anything?
No. I guess just really a special thanks to Alex and Punit, who are the organizers of this year and have given us the chance of innovating a bit on the content and the format of this panel. So I really hope it's been super useful and insightful for the audience today.
Sorry, we have one last question. Can we squeeze it in?
Let's do it. Sorry, Sasha from IDT DNA. Question to for GSK and the venture capitalists. If you have a 75% failure rate, how does that favour with the investors? And throwing that over to GSK, do you mitigate your risks of failure before at such a high rate?
The way that we try and mitigate those is, it may be through, so one way that we can do that is through the financial terms of an agreement. So if there is a lot of risk, then we may include milestones in the deal that are paid after some of those have been de-risked, right? So that's one way to do it. That's the way that we might think about through the kind of deal structure.
From a venture capital perspective, they do a couple things. If it's the single company, right, it's who they hire, who they put around the team that are seasoned experts, that have been there and done it before. They've really de-risked the science. They've really called everybody they can think of to really get an evaluation on, is it a good idea or not, and how much money they're going to put on initially, and how much does it need, right, to really ensure that you can really get to a no-go decision on it. Sometimes a little bit, and sometimes that's a lot. I think overall, generally, the next question would be, how do they do that and mitigate failure? And that's really thinking about a portfolio, right? And how do you take companies that might sit in the RNA therapeutic space, but also sit in immuno-onc or sit in some other rare disease or an antibody company, so they have breadth across their portfolio, so they're not heavily invested in one area.
There's one more second part of that question, if that's okay for you. So there's some of the companies that fail, and you talk about repurposing. Do you ever look back at those failures and see if there's a synergy to help two companies that have failed actually grow?
Yeah, I think there's a lot of times where they'll have a company that's failed or two companies that have failed, and they'll figure out, or they're failing, right? Or they're not sure what they're going to do, but they might have things that they can bring together that can make them both successful, right? I don't know if you just saw in the news, Laurent and Senda just decided to merge out of both flagship companies, but one has an LNP technology, one has a cyclic mRNA technology, and they both kind of need each other, right? They both provide a nice fit for them to make a joint entity to kind of be two halves of the same whole.
Perfect, thank you.
Excellent. Well, thank you. Thank you to the audience, and thank you to the panelists. I hope you enjoyed. We'll look forward to the rest of the conference. I don't know what happens next. Do you have anything? Oh, food.
I was in the shower. I'll call you in an hour. You want to come out now? Sitting there, nobody makes me feel quite the way I feel about you.